Okay, so we're continuing and we're talking about the template strand and how the base pairing mechanism and principle makes one strand automatically the mirror to the other strand, to the anti-parallel strand. Um, so the template strand can be synthesized into what we call a daughter strand. The daughter strand is the complementary strand to the template strand, right? So in the daughter strand, where we have a C, it means that in the template strand we had a G. Where we have a T, we had in the template strand an A. Okay, so it's a very simple idea. If we pair the template strand with the complementary bases, we are producing a duplicate of the original, right, parental strand with great accuracy. Every parental double helix DNA molecule will produce two daughter strands. Why? Because it has two strands to start with, right? The double helix DNA has two anti-parallel strands that are connected by hydrogen bonds. You separate those two strands, and now each one can be used as a template to accurately synthesize one daughter strand. Okay. Let's see. If it all goes correctly without any mistakes, the sequence is preserved. So the daughter strand is often regarded as a hybrid strand because it has been produced by one half of the parental DNA molecule. In the process of replicating the parental molecule, hybrid strands of different weight might be produced. Okay. The fact that after this first cycle of replication, one strand is a daughter strand makes this form of replication what we call a semi-conservative replication. Right? That's because only half originates from the first DNA, let's call it original DNA molecule. The medium in which the strand was grown affects the weight of the strand produced, meaning that different areas of the cell might influence the weight of the strand. We're not going to go into exactly the mechanisms that are going to be affected and how exactly does the weight change. The important part right now is the understanding of an original strand, a hybrid strand, the whole idea of daughter strands, and that the sequence has to be kept when something is replicated. So you are referred to a movie, enjoy, and when we're talking about replication, we have two strands, okay? I'll move it over here. Let's say they are connected, although they're not really, you have the base pairs in the middle, right, with the hydrogen bonds, but just for the illustration, let's say you are connected, and when you start breaking or untying the hydrogen bonds between the bases, it opens up. This part is what is called a replication fork. The fork is the area where the hydrogen bonds are being separated, okay, and we're starting to achieve two single non-connected strands. Okay. So I guess that you already know that we use enzymes to do almost everything in the body. So we have task-specific enzymes that are responsible for the production of the daughter strands. Right? The enzymes specialize in attaching nucleotides to the parental strands, meaning what we're calling pairing complementary bases. Okay, so enzymes are the ones that are putting for every A that we have a T or a U, and for every G a C. Okay, these enzymes are divided, at least we won't go any more further into it. So we have DNA polymerases and RNA polymerases, meaning those who act upon DNA and those who act upon RNA. The DNA polymerases are enzymes that specialize in the synthesis of DNA strands, meaning daughter strands, the replication of DNA. RNA polymerases, as you can understand, are enzymes that specialize in the synthesis of RNA strands. In a similar way, if we want to degrade a molecule, we're going to have one for DNA and one for RNA. One is called DNases and one is called RNases. Okay. We can take the nucleases and also divide them into exonucleases and endonucleases. So the endo ones can separate bonds in the DNA and RNA molecules, and some can operate on a single strand and other on both strands at the same time. So there's different efficiencies and possibilities with them. What happens when these use is that they kind of cut them into pieces. So imagine you have a thread or a piece of paper that's going to a certain length, and you have scissors and you're just snipping. Alright? So those are the endonucleases. The exonucleases move residues, meaning they're taking parts off from the sequences, but only one at a time. So they're very, very specific. Okay? And it's always from the end of the molecule. That means either from the 5' or the 3', right? And what we have when exonucleases work is the mononucleotides. So they're taking out nucleotides, single nucleotides. That's what they're doing. They can operate once again on a single strand and are very direction specific, meaning that they are exonucleosides that work on the 3' end, okay, and they're those who work on the 5' end. If you have one exonuclease that works on the 3' end, it won't be able to work on the 5'. Does that make sense? Okay. So we refer to a movie. Actually, 2 movies. Enjoyed them both. Once again, we're talking about the mechanism of base pairing, which is very simple and has a few options. So we're talking about 3 options. We have the intramolecular pairing, which happens in RNA. We have the intramolecular pairing of different lengths. So we'll see that in RNA too. We'll actually talk about it in the future. And we have DNA. Okay, so right now we're in the cell, and we have 2 complementary RNA strands. Okay? They can be combined, as you can see in figure 1.16 in the middle. And if these two RNA strands are combined and they complement each other, that is what we call intermolecular pairing process. Okay? That means that those strands are complementary to each other and they can simply be binded together. Okay? If we have two strands that are not the same in length but can be combined in parts of them, that's what we call the intermolecular pairing length process. Okay, and then the parts that are complementary can be cleaved by enzymes and used elsewhere. So it's kind of the idea of used parts. So you have a car. Some of the parts are still functional and can be used in a different car that you want to repair. So you're just taking bits and parts from that one and using it in a more functional way in a different place. Okay. Since the base pairing is so easy, and once again, A with T or U, right, and C with G, that means that we can also pair DNA and RNA because it's the same mechanism. Once the hydrogen bonds are separated, as we said, the strands simply go their own way, but it also means that if we establish a new hydrogen bond between complementary strands, they can be reunited. Okay, so it's like a puzzle. Let's say you got a puzzle and it was already made in the box that it was already done in the box that you have. Now you're taking upon the parts that complement each other that make the puzzle, right? But exactly like you took them apart, you can now put them back. That's the exact idea of base pairing. Okay. Let's see. So, how do we separate DNA in a lab? We're not going to learn how to do it exactly, and we're not going to exercise it. But any non-covalent bonds that we have within a molecule, if we start heating the strands, they'll break up. Okay. Another way to break it up is to expose that strand, that molecule, to low salt concentration, and those bonds will just separate. So that's how, in a lab, for instance, we can take a DNA molecule and separate the two strands. Okay. The process of that separation through salt or a low concentration salt and heat is what we call denaturation or melting. Okay? Because you are melting the bonds between the bases. Different molecules of DNA are made out of different sequences, right? So they will have a different combination of the bases. Because do you remember we said that it can go from 24 to about 74, 26 to about 74%. And the combination of the different bases is going to change the amount of energy that we have to invest in the process in order to separate the strands. Temperature is one example for energy, right? So we have a different temperature range, for every melting process. So every molecule has its own melting temperature that we abbreviate as Tm. Okay. The more G's and C's that you have in a molecule, the more stable the molecule becomes. If a molecule is more stable, you're going to have to invest more energy, meaning produce a higher temperature to have it melt. Okay? So we can see that in most mammals, and we are part of the mammal family, that will be about 85 degrees Fahrenheit, which is equivalent to 30 degrees Celsius. And that will indicate that about 40% of their genome are G and C pairs. It's very important to understand that we don't always want to break up the DNA molecules, so the more GCs that you have in a genome, the more stable the molecule, the less it's going to break up. Alright, we're going to stop here, continue in the next video. See you soon.